Hello, everyone, and welcome to our keynote presentation. This webinar is part of the 15th annual event in the Clinical Diagnostics and Research Virtual Event Series brought to you by LabRoots. I am Israel Gonzalez of LabRoots, and I will be your moderator for today's presentation. On your screen are several interactive windows, and we encourage you to engage with them during the presentation. You can speak with other attendees in the Chat with Your Peers window, and also submit questions for our speaker using the question submission window on the bottom right. You may also receive free continuing education credits by clicking the link in the abstract window. I would now like to introduce our keynote speaker, Dr. Xuehai Wang, Scientific Director, Clinical Flow Cytometry Lab at BC Center. To view Dr. Wang's complete biography, click on his name found on the left. Here to give his presentation titled Automating Data Analysis in Clinical Flow Cytometry, here is Dr. Xuehai Wang.
Great. Thank you for the invitation. I'm truly grateful for this opportunity for this talk. I'm going to share our team's experience with machine assistant approach for automating the data analysis in the clinical flow cytometry lab. So here's an overview of today's presentation. I'll begin with a brief introduction to the clinical flow cytometry lab in Vancouver, BC, Canada. So that's where I came from. And I will then move into the core topics, flow cytometry data analysis, automation strategy, and the essential tools and the modules involved to build automated data analysis pipelines. And next, I will highlight two projects to illustrate how we build this machine assistant analysis pipeline to tailor the clinical and the research needs. And finally, I will conclude my talk with a future direction for my lab. So first, clinical flow cytometry in the lab. So just for you guys who is not familiar with flow cytometry, so flow cytometry is a very powerful tool for measuring and evaluating the immune phenotypes in individual cells. So here is a simplified version flow cytometry workflow. So we first label the antigens, often cluster of differentiations called CD markers, with fluorochrome through the antigen-antibody reactions. So these labeled cells are then analyzed by the flow cytometry, which detects intensity of each fluorochrome on every single cells. These fluorescence intensity correspondent to the antigen density on the cells, allowing us to identify the cell types, characterize their immune phenotypes, and quantify their proportion within a given samples. So in the clinical settings, we use flow cytometry to the first enumerating the cell numbers, such as counting the helper CD4 T helper cells in the HIV infected patient. And also we can identify blood cancer cells. So flow cytometry is essentially a very important tool to identify leukemia and lymphoma cells based on their abnormal immune phenotypes. And it can also further divide these leukemia and lymphoma cells into distinct immune subgroups, each of which often requires a drastically different treatment. And flow cytometry also play a very important role in immune monitoring, for example, monitoring the CAR T cells during the CAR T immunotherapy. So in BC, the most clinical flow tests were conducted by three major hospitals, Children's Hospital, Vancouver General Hospital, and the BC Cancer. So each one does a set of tests. There's no overlapping between them. So the patient sample were routed like different hospitals based on their test types. The BC Cancer and the Vancouver General Hospital handle specimens from patients with LPD, which is short for lymphoproliferative disorders, AML, which is acute myeloid leukemia, and multiple myeloma. So we are processing around 6,000 to 7,000 samples annually. So here are the recent developments in the clinical flow cytometry lab at BC Cancer and VGH. So since 2014, we have been using a 13-color 3-tube assay for LPD diagnosis. and upgrade to a 14-color assay in 2020. And we also updated our assay for acute leukemia and acute myeloid leukemia minimal residual disease test from 8-color 5-tube assay to 12-color 5-tube assays. And currently, we're testing a 32-color 2-tube LPD assay expect to be operational early next year. So apart from this development of clinical high parameter flow cytometry, we are also an early adopter for automating the data analysis in the clinical lab. So in 2020, which is 4 years ago, we developed and implemented an automated program to replace the manual analysis for all the LPD cases in the clinical flow lab at BC Cancer. All right, so let's dive into the main topic, how to automate the flow cytometry analysis in the clinical lab. So the first question is, what do we need for this task? I think it is like more with the pre-made bricks. So these modules include data import, export, compensation, data transformation, clustering, classification, dimensionality reduction, and finally, result visualization and GUIs. So I'll focus in this presentation on introducing the automated gating, clustering, classification, and the dimensionality reduction modules. I don't think I will have enough time to go over the data pre-processing, visualization, and the GUI part, which are also very important, especially system in the clinical lab. We can have another separate talk focus on those if you guys are interested. So when we analyze flow cytometry data manually, we mainly deal with 2D scattered plots, like I showed here. Have you ever wondered where these plots come from? So these 2D plots are derived from a matrix like the one I showed here. So each row here represents a data point, usually a cells, and each column records the signal intensity of fluorescence marker, determining the event's position on the corresponding axis. So the 2D plots are based on a pair of selected feature or column. It is important to know that these matrix structure form the basis for applying machine learning and statistical learning and all the different automating algorithms to analyze the data. And the second important thing is to mention that the flow cytometry data is intrinsically high dimensional. So we can approach the high dimensional data analysis from either low dimensional or high dimensional perspectives. For example, let's look at this. Suppose we have flow cytometry only measuring 3 markers, CD3, CD4, and CD8. So these 3 parameters form a 3D data space. So those black dots here in the middle represent the cells distributed in the 3D space. And the gray dots on the side and bottom are projection of these cells onto the different standard 2D plane or surface. So since our computer screen is 2D as well, so we can actually draw lines to divide those cells into different cell groups, or draw the gate to select specified group of cells. This is what we usually do in our manual analysis. So simulating manual analysis, we can also teach the computer to identify the data distribution patterns on a 2D or 1D projection, and then do the gating. So another approach is, instead of dealing shadows or projection of real data, we analyze the high dimension data in high dimension space. This approach is initially applied to CyTOF data. So perform gating or selecting cell population in high dimensional space is almost impossible. Just imagine you try to gate and select the cluster in a 3D space. So for this type of approach, we rely on algorithms to deal with, such as clustering or classification algorithm. So clustering groups data points are here. So we can class the data based on phenotypes into different groups of cells, represented by different color. And with a classification algorithm, we can identify cell types and a group. So here we can identify it as CD8 T cells, CD4 T cell, monocytes, and B cells. So an intermediate method, which is gaining popularity recently, is called dimensionality reduction. It projects or like reconstructs the high-dimensional data into a low-dimensional space, usually a 2D space. So using our 3D color flow, 3D color flow, for example, the algorithm will reduce the 3D data into a 2D plane here. So once we reduce this to 2D, then we can apply traditional gating on this in this dimension reduction plot. So I'll use the following couple slides to go over this algorithm one by one. So let's first look at automated gating on 2D scatter plot. So this method is invented by Ryan Brink's lab here at BC Cancer. So when an expert, a human expert, performs the gating, so we always place a gate based on the density distribution of the events in 1D or 2-dimensional space. So these plots show a granular size and lymphocytes forming two different density peaks on either 45, CD45, or side scatter plot. So the gates are usually placed in the valley between these two peaks. So using this principle, we can let the algorithm to fit a density function to the data and find the peaks and valleys by calculating the first or the second derivative of this function to find the location to place a gate. Unlike sequential manual gating, we can build a pipeline to automatically gate a series of scatter plots step by step. The main advantage of this method is its simplicity. It's very easy to interpret because it's just mimic how human approach data and how human manually gating the data. However, sometimes it lacks flexibility, especially when dealing with unusual cell population and unusual cell density distributions. And then we have the unsupervised clustering algorithm. So flow cytometry data analysis often cluster-based. So we never make a conclusion or diagnosis based on a single data point, but rather the group of similar cells, like we showed here. So manual gating can be considered as a form of clustering analysis. So our brain automatically identifies cell groups from these 2D plots. However, for higher dimensional data, clustering algorithms are necessary. Let's look at the dimension reduction, this DR plot for CyTOF data, so which include 190 examples and measuring 38 parameters or markers on each single cells. So here include 36 normal lymph node samples and 154 flickering lymphoma samples. So if you do a mental clustering in 38-dimensional space, it's just impossible. But with the help of a clustering algorithm, you can group these cells into 38 different subgroup cell subgroup based on all the information from 38-dimensional space. So the common clustering algorithm include these, like k-mean, flow peaks, flow sum, and recently I think flow sum and phenograph is really popular among the algorithm that we use for analyzing or clustering the flow cytometry data. So a challenge with clustering analysis is that the clustering can only group the cells, but doesn't really identify the cell types. So to identify the cell types, that's a job or like task of a classification algorithm. So the classification algorithm usually involves supervised learning, require label data training to let the model learn the rules to put the cells into different categories. So again, for simple tasks like this, telling an identity of CD3 positive and CD19 negative cells is easy. It's a very straightforward classification question. But returning to the previous CyTOF examples with 38 different parameters, we can train the classification models based on all the markers, and then the classifier can use all the information to identify the cell cluster into different B cell subtypes. So for here, we can identify naive B, memory, GC, plasmablast, plasma cells, based on the 38-dimensional space. So classification algorithm can also extend from cell level classifier to sample level classifier, determine if a sample is MRD negative or positive, or identifying those lymphoma cells, which the subtype of those lymphoma cells is follicular lymphoma or DLBCL. So here I listed a couple of common classifiers. So currently, I think Random Forest and XGBoost are very popular one among the single cell studies. So next, we will discuss the dimensionality reduction, a very practical data visualization method. So I believe you guys already saw this UMAP or t-SNE plot many times. But in the next slides, I want to provide an intuitive way for understanding the concept and pitfalls of this dimension reduction algorithm. So let's start with the image from the left. So this is a 3D marmer skeleton. So in manual analysis, we are analyzing data projection by switching the combination of axes. So we can only switching from the 2D plane. This guy, this, or upside down. So you might wonder how this is related to dimension reduction. So, but the most classical linear dimension reduction algorithm, PCA, which is called principal component analysis. You can think of it as how the PCA works as just rotating the data until you find a new viewing angle, the best describing the data. In another word, PCA does the dimension reduction by finding the best projection angle. It doesn't really alter the data structure. So in contrast, let's look at this nonlinear DR method called like UMAP or t-SNE. So those algorithms reconstruct the high dimensional data in low dimensional space and trying to find a version of reconstruction that has smallest difference between the original and low dimensional space data. So this nonlinear DR method is very effective to unfold the nonlinear structure in the data, but this procedure also can introduce potential distortions. For example, let's look at again the mummers. You can see the mummer's leg is disconnecting the UMAP plot, and so the lesson here is one should not over-interpreting the distance in the UMAP space. And secondly, the parameters in the nonlinear dimension reduction algorithm is very important. So changing the parameter will affect the DR plot dramatically. For example, the NN parameter, which is nearest neighbors. If this parameter defines the size of data neighborhood, it gives the weight whether to emphasize local or global structure. So in UMAP, you have it's called a nearest neighbor, and in t-SNE, it often calls the perplexity score. So if I really reduce the nearest neighbor to 3, and you can see the UMAP failed to reconstruct the useful representation of the mummers. So this slide summarizes automated analysis modules and strategy we've discussed, comparing manual versus auto analysis. So the automated analysis provides a great freedom of flexibility, enabling the integration of automated 2D gating, finding suspicious population by clustering, optimize the data visualization, and offering customized functionality beyond those provided by commercial softwares. So going back to the Lego analogy, building an automated analysis system is like assembling Lego bricks into various structures. architectures. So here are several architectures developed or used by our and other groups. So first is AutoFlow. It's our LPD automated analyze system. It integrates the clustering and the dimension reduction following the automated gating steps. And this is our MagicDR. So this is our AMR MRD analyze system. So using dimension reduction and single cell level classification. So other teams structure performing clustering in dimension reduced space, or combining clustering with classification analysis. Here are some simplest architecture. However, the simplicity does not necessarily mean the system performance is not as good. So there's a recent study put out from Mayo Clinic highlights the neural network classifier for uncompensated data. So eliminating this compensation would greatly simplify the workflow. And this slide lists the open source flow cytometry tools based on R and Python. Because these languages offer a wide range of packages handling biological data. We use these packages as shown here, the Flowco and the Flow Workspace for data import, export, and pre-processing, and Flow Density for automated gating analysis, ScanP and FlowSum for dimensionality reduction and clustering, and lastly, the RShiny for the GUI, and also Flex dashboard for the GUI. So having understand the flow cytometry data and some commonly used analysis module, I now want to share how we use this automation and AI to improve the efficiency of the lab workflow. So this was our original workflow in the flow cytometry lab. So in our lab, the flow data was mainly analyzed by the technologists. The result got exported as PDF using the commercial software, export result PDF, and then was sent to the pathologist. If there were questions, the pathologist will communicate with technologist for adjusting or reanalysis. And the pathologist will using the PDF to issue the report, diagnosis report. So here is a plot to identify which tests we would like to prioritize for the automation. So lymphoma and AMR-MRD are the most complex. The text volume also for the LPD is quite high to our current lab size, and the volume is increased year over year. And so definitely this will be the top pick. But on the other hand, we don't receive too many AMR-MRD tests compared to the LPDs, but AMR-MRD analysis is one of the most, if not the most difficult data analysis task in the clinical flow cytometry lab. So we would like to come up with something to simplify the analysis. So we name our two programs as AutoFlow and MagicDR. So for AutoFlow, we build it as a standalone software. So it generates analysis report for pathologists without any manual intervention. So basically, it takes FCS file directly from the machine of flow cytometer and generates report. For MagicDR, we designed for better data visualization and highlight suspicious cells that bearing abnormal phenotypes. So the results from MagicDR need to be loaded onto traditional commercial software for further analysis. So one thing I would like to emphasize is that our approach of machine learning or machine assisted analysis pipeline is to help or assist the diagnosis instead of making decision for you. Okay, so the first system is AutoFlow, as for LPD diagnosis. So first, I will just briefly talk about the LPD flow cytometry panels currently used in the DC Cancer Flow Lab. So here we use a 3-tube, 14-color LPD panel. So we have 2 B tube and 1 T tube. So for the tube B1 contains kappa and lambda light chain antibody, and also CD10 and CD5, so as to identify the B cell clonality and also for B cell phenotyping. And the B2 tube is kind of complement to B1, including markers such as CD23, CD200, CD103 for further B-cell lymphoma subtype identification. So for T-tube, the T-tube contains markers for T-cell lymphomas. And also we have TRBC1, which is introduced 4 years ago to identify the clonal T cells. So first, I will start with how to use supervised automated gating for the T-tube. So for T-tube automated analysis, we try to mimic the manual gating. We're using the density-based automated gating strategy. So first, we apply a gate to select the lymphocytes and the monocytes as our expanded lymph gating. Then we remove the monocytes by CD14 versus CD45 IgM plot, and followed by selecting T cell based on CD2 and CD3, and subgate this into CD4 T cell and CD8 T cells. And then we can check their CD5, CD7, TRBC1, RA expression on different T cell population. So the automated gating is very fast and efficient. It can generate over 300 subgroups in less than 20 seconds. Here we only color the major population as shown in the right figure. So major subgroup are colored for easy observation, and the stats of these subgroups, ratios, and percentages within the sample are also automatically calculated. So the same analysis strategy doesn't really work well for the B-cell tube due to the B-cell heterogeneity. So CD20 and CD19 expression can vary, especially in the lymphoma cases. And also lymphoma sample often contain both normal and tumor cells, complicating the analysis. So for B cells, we use a different approach, a hybrid approach to analyze the TB. So first, we use a logical OR gate to label this. So we label the cells positive for either CD19 or CD20 as B cells. And then we can apply a clustering algorithm to these B cells to separate potential B cell subsets, as well as separate abnormal cells from normal cells. Additionally, we use dimensionality reduction algorithm. Here we use UMAP to display the relationship between B cells versus monocytes and T cells, and also display the relationships among different B cell subgroups. So the other unique feature of our autoflow algorithm is the report are delivered in web HTML format instead of PDF. And the web HTML format provide a much improved user experience. So we can see the header shows the sample ID, tissue type, and also options to switch between results from different tubes. So instead of like static PDF files, for HTML file, we can click different tabs to bring out different results from different tubes. And let's focus on the B1 tube. So the left side show all the stats, the proportion and the ratio are shown here. And the middle part showing the initial gating steps and UMAP plots for the major lymphocyte population. And the detailed immune phenotype are displayed in traditional bivariate scatter plot on the right. The UMAP plot is actually interactive. You can interact with UMAP plot. So we can, let's say, zoom into this B cell population. It shows 4 different B cell subgroups, each represented by different shades of blue. So these colors match those in the traditional 2D plots on the right. So from the traditional 2D plot, we can see the B2 population is a polyclonal B cells based on the it has both kappa and the lambda population, while the other B cell subgroup are kappa restricted. So you can see that only express kappa. On UMAP plot again, so B2 population B2 is a bit shifted from the B1 and B3 population because B2 is normal B cell population. And interestingly, the B4 is also a bit shifted away from the B1 and B3 island. We can click the B4, BPOP4, for further interrogation of this B4 immune phenotype. So it brings out this detailed immune phenotype. So we can see the difference between B4 to B1, B3. If we click the B1 and B3, we look at this CD5 expression that we can see B4 is different from the rest because of CD5 positivities. And next, one could be wondering the difference between B1 and B3 population. Again, by clicking the B1 tab or B3 tab, we can actually see that they differ based on their CD20 expression. So B1 has slightly increased or like slightly normal CD20 expression than B3, which is almost CD20 negative. So here I want to emphasize again, the main idea behind AutoFlow is to highlight these different B cell population, extract and present useful information from the raw FCS flow cytometry data, and the decision on how to interpret this information is up to the pathologist. For example, in this case, so we can all agree that there are kappa restricted B cell population, which is B1, B3, and B4. And some of them express E5, which is B4 population. But whether you would mention the residual normal B cells as well as to a different population within different intensity of CD20 is up to the pathologist. So similarly, on the T cell page, the middle column shows how the gating strategy for T and NK cells. And left, again, it's the stats panel, and the right is displaying the detailed phenotypes for CD4 and the CD8 population, including the TRBC1, CD5, CD7, and RA and RO patterns. Okay, so starting autoflow analysis is very simple. So this is the page usually stay in the lab for the lab technologist. So there's only two steps. So first, we click load the files, and the second step is click start. Then you can just walk away for a coffee. It takes about 3 to 5 minutes for analyzing one case, including one tube B1, B2, and T3 tube altogether. So AutoFlow has been operational since 2020, so it's like 4 years ago. So it replaced the manual analysis and eliminating the inter-analyst variations, and more importantly, also saving at least 1 FTE. Okay, so the next is MeshDR, which is a machine learning approach to help our AML-MRD analysis. So here's our AML-MRD panel. Same thing as the LPD. So first is a panel. So we use tube 1, 2, 3, and the first 3 tube as our AML-MRD panel. So tube 1 and 2 include some early differentiation markers like C38, HADR, 1333, and also lineage markers like CD7, CD2 on the tube 1 in tube 1, and CD19, 22, CD10 on in tube 2. And tube 3 include monocyte maturation markers, so which is very specific for monocytic differentiation type of AMLs. So the MeshDR consists of classifiers and the dimensionality reduction module. So the classifier will give each cell a tumor probability or AML probability score. and the dimensionality reduction module perform UMAP analysis. So one feature of the Magic DR is that data, including the probability score and the UMAP coordinates, will be right back to the original FCS file to create an enhanced or augmented FCS file. So these files are just like regular FCS files, can be loaded onto any commercial available data analysis software to visualize the tumor probability scores in UMAP space. So for dimensionality reduction, we choose UMAP for two reasons. So first, it's very effective in visualizing continuous data and the nonlinear structures, which often presents in change of phenotypes during the immature cell development. And the second is it's very fast, especially compared to t-SNE. So here's a comparison between these traditional bivariate plots and the UMAP on the population from on an example from post-induction marrow. So the UMAP often untangles this complex data into a very intuitive two-dimensional plot, separating all the maturation pathways of different cell lineage from each other. So here is for neutral cell or granular size differentiation. Here is for the monocytes, erythroid, and B cells. So the AML classifier were trained with cells from over 100 cases, including 30 AML MRD negative samples and 98 different AML cell population from over 80 patients. So we combine all the data into a large matrix and add a column to indicate whether the cells in each row are normal or leukemic. So the previous studies reported building similar classifier, but after the clustering or on a dimension-reduced space. We decided to build a classifier on the single cell level. So this is because for AML MRD analysis, we are often dealing with very small number of cells, and the percentage of MRD population in AML MRD sample could go as low as 0.1%. So when dealing with this tiny percent of cells, it can be easily masked during the clustering or DR steps. So that's why we decide to just build it at a single cell level. So we're gonna give a score for every cells from the unknown sample. Here we use a very popular algorithm called XGBoost. So AML is also known for its heterogeneity. So that's why we try to include as many training data as possible, including different AML subtype defined by WHOs. Here the table shows the sample count per subtypes, and also the UMAP here illustrates the difference in immune phenotypes among all these WHO subtypes. We use leave one out cross-validation to evaluate the model performance. Here, the y-axis represents AUC score. The higher the AUC score, the better the model performs. So the results show that the performance of our classifier are consistent across WHO subtypes, but varies among the different immune phenotypes group. So our model are doing best detecting CD34 negative blast across all three tubes. You can see we can achieve very high AUC score, and also the cluster is very tight. While monocytic AML cases have better chance to be picked up by classifier based on tube 3 data, which is here. It's kind of understandable. And the XGBoost also provides information about the contribution of each markers in the classifiers, highlighting the importance of this marker in predicting the AML cells. So in this figure, the x-axis represents different markers in different tubes, and the y-axis shows a number of models in which each marker has a significant contribution. So the message here is that CD117 actually performed unexpectedly well, and is kind of top-ranked marker across all different across 3 tubes. So they perform very well, especially in tube 1 and tube 2. So a side point here is it seems for majority of AML phenotypes, 6 color flow might be sufficient to pick the AML residue cells. And here, this figure is about a dry experiment we did to assess the effectiveness of UMAP in distinguished AML cells with different immune phenotype. So the first row are UMAP plots with normal and abnormal population colored based on the probability score calculated by the classifier. So the color close to red means higher probability being an AML cell, AML blasts. The second row are the same sample, same data, but colored by the true identity. So the color of red means leukemic blast, leukemic cells, and blue means normal cells. So let's look at the first data sets. So the first data sets including cells with a unique, easily distinguishable immune phenotypes. When we had a high number of cells, which given 6,000 cells, the UMAP can clearly separate the AML cells from normal cells. So even when we reduce from 6,000 to 250 cells, the UMAP can still manage to distinguish those AML blasts from the normal cells. So the classifier also have a perfect matched prediction to the ground truth, indicating both UMAP and the classifier is very effective in this scenario. So the second data is a bit different. So the second data set contains the cells with a phenotype not as distinct. So you can see when the population size are big, which is 6,000 cells, UMAP can manage to separate AML bloods, but with some difficulties. When the population reduced from 6,000 to 250 cells, which is 1% of total population, total cells numbers, UMAP's effectiveness decreased significantly, so it can merely pull this population out from the normal territories. But despite this, the single cell classifier remain very accurate. So you can see those cells are labeled as red. It performed very well even in this very challenging condition. So the conclusion here is in dealing with AML MRD cases with very low number of residual cells, UMAP could be struggled, but the single cell classifier is always, the performance is always consistent. So the consistent performance of the single cell classifier highlights the necessity of combining both UMAP and the single cell classification for accurate identification of AML MRD. So this slide shows how the visualization was done using the enhanced FCS file in the commercial software. Here we use FlowJo. So the enhanced or augmented flow cytometry generated by MatrixDR pipeline are imported into the FlowJo, and we can then plot the UMAP plot and along with this traditional bivariate plots using FlowJo. And we can also use the color access function to display the probability score, providing a dual perspective for the analysis. So in example 1, which is a negative sample, MRD negative sample, so UMAP plots show no red dots. So, but in the positive samples, which is AML MRD positive, okay, UMAP plots show a distinct cluster of red dots, identifying cell likely to be the AML MRD cells. And to look at the Biography plot, we can also see a small cluster of red dots here, and there, with C38 dim, 417 high, and C7 positive phenotype. So to conclude the MatchDR models, so integrating a supervised classifier with unsupervised dimensionality reduction offer a simplified approach that can be seamlessly integrated into the conventional workflows. You can use it in any commercial software. And our approach also provide a support and our approach can support and augment human analysis by highlighting this abnormal population that can be gated on for qualification and also for further investigations. So the next will be the future workflow. So as mentioned earlier, we are trying to update our current 14-color 3Q panels to a 32-color panel based on the BD Symphony A5SC cytometer, which is this guy here. And we are also experimenting different options of sample prep robots, just to try to automate all the pipetting and cocktailing part. So it's a wet lab part. And more exciting things is about building the AutoFlow 4.0, which will be based on a panel agnostic model. So this AutoFlow 4.0 will also providing a more interactive functionality like live gating. So we are very excited to developing this platform. So with that, I would like to conclude my talk. Thank you very much. I have time for the questions.
Well, thank you, Xuehai, very much for that informative presentation. Let's go ahead and dive into the Q&A now. And to our audience, remember that you can submit your questions below for our speaker. Let's go ahead and dive into the first question here. What is the most challenging part of this project?
Thank you for the question. It's a very good question. I think there's a couple of things. So first of all, it's very different to design or implement automated analysis pipeline in the clinical situation. So it's quite different from what we're doing here than doing a research paper. So there's a lot of things to consider, not only to select all these different algorithms to put them together as a package, and also have to listen to the needs of the clinical team, what the pathology group need, what kind of format the data will be presented, and what kind of key information will be presented in the report. It's kind of funny to know that when we first generation of this algorithm, we present in a more like a research format, but that's very disorienting. So in the end, the pathologist will actually need a more transparent, non-black box way to show the data that analyzed by the machine instead of a human. So we end up doing a kind of like a hybrid method, as I mentioned earlier, that try to mimic how the human gate and then try to also lay on top of a little bit machine learning algorithm. So I think every clinical lab on even every lab is quite different on what they actually need the algorithm to be. So to me, how the communication between me and the team with the clinical team and also informatics team is quite challenging. Try to understand the need from both sides and try to speak the same language. It's quite challenging. And all these putting the algorithm together part is actually really relatively simple because all these modules can be found online. Yeah.
Okay, wonderful. Looks like we only have time for just one last question here. Can the tool be used in a different lab?
Currently, yeah, it's also like fantastic question. Like I think the currently like with the tools can only applied in our lab with very specific panel design because this is kind of custom built for our panel. So we have to acknowledge the flow cytometry data, flow cytometry assay is quite unique to each the lab. So especially in North America. So in Europe, they have Euroflow can have a shared or like a unified panel for different labs. So which means the data structure, or not data structure, but the data receiving collected in different labs can crosstalk to each other. But in North America, in US and Canada, the situation is quite different. So in BC, we're using 14 color 3-tube assay for LPD diagnosis. But in other places, they might use different 2-tube assay, 8-color 2-tube assay, or 8-color 5-tube assay. And the choose of the fluorochrome for each antibody, and how many antibody, like what different combination of antibody included in the panel. All those things will directly affect your model. But as I mentioned in the future direction, we are currently actively working on a model panel agnostic approach. So which means the next generation of our flow could be applied, or hopefully could be applied to all different panels. As long as you are detecting, as long as you are the panel for the LPD detection, LPD diagnosis, and hopefully the AutoFlow 4.0 will be able to analyze and generate results.
Wonderful. Thank you so much for that. And before we wrap up, Dr. Wang, do you have any final comments you'd like to share today with our audience?
So yeah, I think it's for me, I'm always fascinated by flow cytometry and also fascinated by the clinical application of it. So I think that the future, the machine learning and the deep learning method will be applied in this in the clinical lab domain for sure, because we're always facing the increasing sample volume as well as the labor shortage. So our vision is to try to build a lab with automation all the way through, from the wet lab part, pipetting, cocktail making, adding antibody, lysis red blood cells, and to the data analysis. So this is, I think, we strongly believe that this is going to be the only way to going forward. And I think next generation of the scientists or technologists or hematologists working in the lab will be the person who can deal with this algorithm and who can deal with these robots instead of only good at pipetting. So I think that's a lot of potential in the future. I wish I can make some friends and collaborations in the audience, and very exciting for going forward. Thanks.
Well, thank you so much, Dr. Wang, for your time today and for your important research. Now, before we go, I'd like to also thank our audience for joining us today. Questions that we did not have time for and those submitted during the on-demand period will be addressed via the contact information provided at the time of registration. This webcast can be viewed on demand for 1 year until November 13th, 2025, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Now, feel free to continue exploring this incredible virtual space, including the poster hall, the LabRoots merch store, the social space, and tune in for other wonderful webinars throughout the entirety of today's event. Again, thanks to our audience and to you, Dr. Wang. Goodbye, everybody.
Thank you very much. Goodbye.